In this session of the video, we are going to talk about the signals, various kinds of signals that are generated from human body, that is biomedical signals. In the earlier lecture, we had talked about how the potential is developed at the cellular level. Okay, so although in this particular unit, we'll be focusing only on the electric signals, that is the bioelectric signals, but just to give you a brief introduction of how many different kinds of signals are actually produced from human body, that we give the term as biomedical signals. So we have about 6 to 7 types of different kinds of biomedical signals that are produced from the human body, and these signals can actually be used to they can be measured and they can be used to find out the physiological state of the body. Okay, so first of all is the bioelectric signals. These signals, they are generated mainly by the nerve cells and the muscle cells. Their basic source is the cell membrane potential, which under certain conditions may be excited to generate an action potential. Okay, the electric field that is generated by the action of many cells, it actually constitutes the bioelectric signal. So it may be ECG, EEG, EMG, that is electrocardiogram, electroencephalogram. Okay, all these are bioelectric signals. They are produced by muscles, brain cells, etc. Then we have bioacoustic signals. These are the signals, they are generated by the flow of blood in the heart, through the heart valves, and the flow of air through the upper and the lower airways in the lungs. So when there is acoustic, basically means sound. Okay, so these are the sound signals that are generated in the human body. So broadly, the reason for producing sound signal is, one is the movement of blood throughout the body. Because of that, high or low flow of the speed of flow of the blood, the nerves, the walls of the vessels, they move. Okay, and so that movement, or that movement of the blood, movement of the air in the lung, in and out of the lungs, all these they produce certain sound. So measuring of the heart sound with the help of a stethoscope is a very, very common activity that we see around ourselves. So this is bioacoustic signal, that is a sound signal coming from human body. Then we have biomechanical signals. These signals originate from some mechanical function of the biological system. They include all types of motion and displacement signals, pressure and flow signals, etc. The movement of the chest wall in accordance with the respiratory activity is one example of this type. Okay, so when you are breathing, there is a lot of movement of muscles taking place. That movement is actually a biomechanical signal. Then we have a biochemical signal. The signals which are obtained as a result of chemical measurements from the living tissue or from the samples that are analyzed in the laboratory, they are all biochemical signals. For example, measuring of the partial pressures of gases in the blood, okay, or maybe the pH, measurement of the pH is, again, it's a chemical signal. Okay, so whenever we are taking the sample from human body into the lab and we are analyzing it, the values or the signals that we are getting from that is actually a biochemical signal. Then we have biomagnetic signals. Okay, very weak magnetic fields are produced by various organs of human body, such as brain, heart, and lungs. Okay, the measurement of these signals, they provide very valuable information, which is not actually available in other types of biosignals. So we have an example that is called magnetoencephalograph. That means this is measuring the magnetic signals that are being originated from the brain. So we have the brain is also, giving out magnetic signals. Then we have biooptical signals. These are generated as a result of optical functions of the biological system, occurring either naturally or induced by the measurement process. Okay, so like pulse sensor, we had seen that we are inducing some light, some optical signals, and then the reflected signal is coming back to us. So these are optical signals. They can give you very valuable information. One very popular example is blood oxygenation. Measurement of blood oxygenation is actually done by biooptical signals. Then we have bioimpedance signals. The impedance of the tissue is a source of important information concerning its composition, blood distribution, blood volume, etc. The measurement of galvanic skin resistance is a typical example of bioimpedance signals. The bioimpedance signal, it is also obtained by injecting a current into the tissue and then measuring the voltage drop that is generated by the particular tissue that is under consideration. So bioimpedance actually means resistance. Okay, so where the skin, human skin, various tissues, etc., they all offer certain resistance to current. Okay, and the resistance varies. The variation in that resistance can actually give us a very valuable information about the state of that tissue, or the state of the skin. So bioimpedance signal is basically the changes in the resistance that are caused in the various tissues and skin cells, etc. So these are some of the some total the various types of biological signals that are produced from human body. But in the further studies, or in this particular unit, we'll be focusing only on bioelectric signals. Right. So coming to the bioelectric signals or the bioelectric potentials, some of the popular ones that we have, the bioelectric potential that we have, they are electrocardiogram, ECG, we all know, electroencephalogram, that is EEG, electromyogram, EMG, electroretinogram, that is ERG, electrooculogram, EOG, electrogastrogram, that is EGG. These are some of the important electrical signals that are coming from the human body, and they are recorded, they are measured to find out very vital statistics of human body. Okay, so we'll see how, first of all, let us see the definition of all these, what they actually are. ECG, of course, very popular. It is electrocardiogram. It records the electrical signals coming from your heart. Now, with each heartbeat, an electrical impulse actually travels through your heart. The basic natural way of your heart functioning is actually generating this ECG. Okay, this wave that is generated, it causes the muscle to squeeze and pump the blood from the heart. Okay, then the sinoatrial node, it sends out an electrical pulse. The upper heart chamber, it contracts. Then in the AV node, it sends an impulse to the ventricles, and the lower heart chamber, it contracts or pumps. So basically what is happening, there is naturally an electrical pulse is being generated in your heart, and that pulse, electrical pulse, is actually making your heart muscles squeeze, and the action of pumping blood is being caused because of that electrical signal, that is being electrical wave that is being produced inside your heart. So when we measure ECG, we actually try to see whether the natural capacity of the heart to generate this electrical pulse is okay or not. Okay, if there is some slight variation in the ECG, that means that your heart is not being able to create generate these electrical pulses, and that creates a disruption in the normal functioning of the heart. So you can see in the figure that we place a number of electrodes at various locations, and these electrodes actually capture the electric signals that are produced by the pumping action of the heart. Then we have electroencephalogram, that is EEG. This is basically the recorded representation of the bioelectric potentials that are generated by the neural activity of the brain. Okay, so when there are the neurons of the brain, basically when they are communicating with each other, when they are undergoing various kinds of activities, some electric potential is being generated over there. Okay, so that particular potential that is being generated, when we record that, that is called an EEG signal, that is electroencephalogram. Okay, so when the brain is working, the cells, they communicate using the electrical signals, and when they do this, they give off electricity. Okay, this it is this electrical activity sometimes which you also called as brain waves, that is picked up by the EEG machine. So again, you can see in the figure that we have a set of electrodes, all through the head, you can see that the patient has a number of electrodes. So many electrodes they are placed because you want to have a localized measurement of the signal coming from some particular area of the brain. Okay, so we place, there are various types of electrodes, various numerous ways in which the EEG can actually be recorded. So EEG actually is recording the electrical activity of the brain, right? When the brain is working, when the neurons, they are communicating with each other, they produce some electric current. So that electric current or the electric activity is recorded with the help of electrodes, and that signal is called an EEG signal. Electromyogram, now it comes to the activity of muscles. The bioelectric potentials, they are associated with muscle activity, is called EMG. Okay, so as we have seen that, at the cellular level itself, the bio potentials are being created. So naturally, your muscles are also made up of cells. So the various activities that are taking place in your muscles when you're working, that is changing the electrical signal from your muscles at every point of time. So when we capture this muscle activity with the help of certain electrodes, the signal that we get is called an EMG signal, or an electromyogram signal. Okay, these are measured at the surface of the body near the muscle of interest, or directly from the muscle by penetrating the skin with the needle electrode. You can see in the figure, we have a needle electrode. Either you can, put it in the vicinity of that particular muscle, or you can directly insert that needle into the muscle. Okay, so the muscle activity, when we are recording it electrically, it becomes EMG. Then we have electro retinogram. This is record of the complex pattern of electric potential obtained from the retina of the eye in response to visual stimulus. That means, whenever visual stimulus means, suppose we may incident certain light on your eyes. So how does the retina actually respond? So even your retina is, responding or creating certain electrical potential. Now again, because we know that at the cell level itself there is a potential being generated, so naturally at various places we can expect some electrical signals to be generated, right? So when we allow certain visual stimulus or some external light, suppose, to fall on your eyes, how the retina is reacting, what pattern of complex what complex pattern of electrical potential is being generated, that is recorded from the retina of the eye, and that is called electroretinogram, or ERG. So that will give you an idea of the functioning of retina. Similarly, for the eyes, we have electrooculogram, that is the measure of variations in the corneal retinal potential as affected by the position and movement of the eye. So the potential between the cornea and retina, as you're moving your eye, when there is a movement of eye taking place, and you're positioning your eye up, down, this in various directions. So because of that movement, what is the potential being developed in your cornea and retina? That is electrooculogram. Okay, then we have electrogac. The last one is electrogastrogram, that is EGG. These are the patterns that are associated with the various movements that are taking place into your gastrointestinal tract. So you can see that there are so many different types of bioelectric potentials being produced within human body, and all these, when we need very specifically designed electrodes to capture these bioelectric potentials, and all these bioelectric potentials are actually going to give us a very very valuable information regarding the various states of human body. So now when we are talking of electric potential, we are talking of electrodes. So the important thing that we all should know something is electrode theory, because whenever you're talking of electrodes, the basic functioning of the electrode has to be understood in terms of electrode theory. So whenever the interface of metallic ions in a solution with their associated metal results in an electric potential, that is called an electrode potential. This also, I think, most of you must have been familiar with this in your earlier classes. You must have studied something called electrolysis. That is, when we have a solution having certain ions in it, and we dip an electrode of that same particular metal. When we are dipping it into the solution containing the metal ions, and we are passing the current through it, then electrolysis takes place. That is, cations and anions are formed, and the positive charge gets accumulated at one end, and the negative charge, or the anion charge, gets accumulated at the second one. So the interface of the metallic ions in the solution with their associated metals. So if you see over here in the figure, the cathode, suppose if this is a metal, the metal ions which are positively charged, they accumulate at the surface of the cathode, right? They are called cations. So this interface of the metallic ions in the solution with the associated metals, they result into certain potential. Obviously, when there is a difference in charge, when the charge is positive and negative charge is being accumulated at particular surface, there will always be a potential developed, and that potential we called as electrode potential. Okay, this potential is actually a result of the difference of diffusion rate of ions into and out of the metal. See, whenever the metal is being inserted in that, the ions can actually, there is a difference in the rate in which ions can come out and go into that metal. So that difference in the diffusion rates is actually giving rise to this potential. Okay, and an equilibrium is produced when a formation of layer of charge at the surface. When there is a at the you can see in the figure, at the layer or the interface boundary of the cathode, when it is covered with the layer of charge, both anode and cathode, then a kind of equilibrium is maintained, and the charges, they get balanced, right? So now, when if we are using a semi-permeable membrane that separates two liquid solutions with different concentrations of a specific ion, an electrode potential will obviously develop. We are having two liquids. Okay, they have different solution, different concentrations of the same ion. Suppose we have liquid A and liquid B. Okay, liquid A has concentration of sodium as x1, and liquid 2 has concentration of sodium as x2. And if they are separated by a semi permeable membrane, then on both the sides there will be an electrode potential will develop. Okay, since it is semi-permeable, means that the ions will not be allowed to move freely through that membrane. Okay, and the concentration is different in both. So if the sodium ions, the liquid which has the higher sodium ions in it, concentration of sodium is high, that will achieve a positive charge, and the other side will be negative as compared to the positive one, right? So a potential electrode potential will develop across this semi permeable membrane. Okay, and this potential that is developed is actually governed by the famous Nernst equation. The potential is equal to minus RT upon NF log of C1 F1 divided by C2 F2, where R is a gas constant, T is the absolute temperature in Kelvin, N is the valence of the ion, F Faraday constant. C1 and C2 are the concentrations of the ions on the two sides of the membrane, and F1 and F2 are the activity coefficients of the ion on the two sides of the membrane. So these are actually dependent upon what are the ions, what substance you are taking into consideration, what is the solution, that decides F1 and F2. So this equation we have to remember. Even in the further lectures, we will be using this equation while, if at all you have to design some electrode for a particular measurement. The Nernst equation is supposed to be remembered, because this actually governs the potential that is going to be developed. So even when we have to measure what potential will be developed, if we know the concentrations, we can actually find out the value of potential developed with the help of the Nernst equation.